In this video, we are going to talk about the retroviral life cycle, taking an example of HIV. Retroviruses. These are a class of animal viruses. They are enveloped viruses. That means they possess envelope towards outer side of capsid. Second thing is their genome consists of single-stranded RNA, which is linear and non-segmented, approximately 7 to 12 kb in length. They have three genes, that is GAG, POL and ENV genes, which have structural role present in all the members of this family. Enzyme reverse transcriptase is characteristic of retroviruses. It has two enzymatic activities. One is polymerase activity and the second one is RNase H activity. We'll look onto these activities in the next half of the video. Another thing, now we'll look on to the general retroviral life cycle and then we'll move on to the specific HIV life cycle. Plus, since RNA is present in the genome of all retroviruses, it acts as template for the synthesis of negative sense DNA strand plus sense RNA. Now, using RNA as template, DNA strand has been synthesized. So this must be a function of RNA dependent DNA polymerization. So this is the polymerase activity of reverse transcriptase enzyme. Then, since this RNA DNA duplex is very unstable, as RNA and DNA cannot be bound for a longer time, so this RNA component needs to be removed. What is the function of RNase H activity in reverse transcriptase is to remove the RNA component of this duplex. Again, we are left with the single strand of DNA. Now, this minus sense DNA will act as template for the synthesis of plus sense DNA. This is DNA dependent DNA synthesis, again using the polymerase activity of reverse transcriptase. Hence, RNA template has single-stranded RNA has been converted into double-stranded DNA with the help of reverse transcriptase known as cDNA, complementary DNA. Now, this double-stranded DNA needs to be integrated into the host chromosome, host double-stranded DNA in the nucleus. So, this is the host DNA with the help of enzyme integrase. We insert or integrate into the host chromosome at random sites, random integration sites. Integrase has three activities again. It has exonuclease activity, endonuclease activity, and ligase activity. Exonuclease activity. Look, this is the 3 5 3 5 N. This is the viral DNA. Now, at the 3 ends of viral DNA, this exonuclease activity will cleave two nucleotides from each 3 end of viral DNA. First thing, so it gives cleaved viral DNA. Endonuclease activity, it will cleave host chromosome from random sites where viral DNA needs to be integrated. So it will give us cleaved host DNA. And again, the function of ligase is to seal. It will seal both the cleaved DNA and will form host plus viral DNA integrated, which is called as provirus. Now this provirus needs to be transcribed. This is the DNA. It needs to be transcribed to encode for proteins. Now its transcription takes place. On transcription, it will synthesize genomic RNA, that means one which is to be packaged in the virus via genomic RNA, and another one is mRNA, which is to code for proteins, viral proteins inside the host. Genomic RNA, it will move to cytoplasm, host cytoplasm, where it will be covered with the capsid proteins synthesized from this mRNA. These capsid proteins will cover it, and From the host membrane, budding will occur and host membrane will convert into envelope in that part, and the capsid will be covered with envelope and released. So this how the complete virus structure is made out of the host and released. Next, we'll discuss HIV in detail for the life cycle. HIV. HIV is human immunodeficiency virus, which has been discovered in 1983 by Montagnier and Gallo. It has been discovered in the patients suffering from lymphadenopathy. Lymphadenopathy. So, the HIV was even formerly called as LAV, lymphadenopathy associated virus, or ARV, AIDS associated retrovirus. Why is it called so? Because it is the causative agent of AIDS, acquired immunodeficiency syndrome, discovered in 1981 itself. The AIDS patients showed the decrease in CDR, CD4 positive T cells. That means T cells having CD4 as marker on them. They were decreased in number in those patients. Now, what makes them decrease in number? We'll see to it. First of all, we'll see that HIV belongs to family retrovirus, and subfamily is lentivirus. Mostly HIV is caused due to HIV-1, AIDS is caused due to HIV-1, and in some cases even HIV-2 is seen to be responsible. Its symmetry can be icosahedral. It is icosahedral symmetry. That means a regular polyhedron icosahedron. Now, let's look on to the structure of HIV. Structure. Outer layer is envelope. Inner layer is capsid. In between both of them is present matrix. Matrix consists of protein. Now, envelope is made up of spikes. Spikes is made up of two segments. First is GP120 and the other is GP41. Now, in this notation, G stands for glyco, P stands for protein, and this numerical figure, it stands for the mass of protein in kilodaltons. This is a general notation we are even going to encounter in the later half. This GP41 is the transmembrane segment, and this GP120 is the external segment. That means if this is the membrane, then within the membrane the part is called transmembrane, and the part protruding outwards is called as the external part. This combined act as viral receptor for CD4 marker on host. Now, this matrix part is consist of P17 protein with the molecular mass of 17 kilodalton. Nucleocapsid is made up of 2000 copies of P24. Now, looking on to the genome of HIV. First of all, let's talk about the three important structural genes. The function of them is GAG gene, Pol gene, and ENV gene. These are the genes which encode for GAG polyprotein. Look, the gene is represented with a small starting from a small capital, whereas small letter, whereas protein is denoted by starting from the capital letter of the same name. ENV encodes for ENV polyprotein, and Pol gene, although it encodes for Pol polyprotein but not directly, it codes for it indirectly. That is, GAG and Pol gene together synthesize GAG Pol fusion protein. And on the breakdown of this fusion protein, we get Pol polyprotein. So there's an indirect way of getting this. Now, Pol polyprotein is further cleaved to synthesize three enzymes: protease, reverse transcriptase, and integrase. This protease is viral protease is very important as it would as it is responsible for cleaving this fusion protein first of all. Then it cleaves this polyprotein into these three enzymes, and third it cleaves GAG polyprotein into three different type of proteins: capsid proteins, matrix proteins, and nucleocapsid proteins. Capsid proteins are also called as P24. This is P17, and this is broken down into two types, P6 and P9. Reverse transcriptase, we have two activities, polymerase activity and RNase H activity we just studied, and there are three activities of integrase: exonuclease, endonuclease, and ligase activity. Now, ENV polyprotein is further cleaved into two parts. We have just studied that envelope is made up of GP41 and GP120. But the different thing here is that it is cleaved by a host protease, whereas all other cleavages in this has been done by viral protease. Now, with this, we are clear on this topic, and we'll discuss about the mRNA, which has been synthesized here, its transport. mRNA is synthesized in the form of primary transcript. Now there's a mechanism how this is spliced and transported to cytoplasm. The genes, there are 9 genes present in the viral RNA, and there are some terminal repeats in both the ends of this genome, this RNA. These are long terminal repeats. That is, a repeat sequence is present at both the ends, and there are 9 genes in between, naming them GAG, POL, and ENB. These three are common, but additional genes are VIF, VPU, VPR, TAT, REV, and NEF. Now, when this is encoded by the, we have just seen that when it is encoded here, the mRNA is synthesized on transcription. This mRNA is synthesized in the form of primary transcript. Now, this primary transcript, it may be spliced or it may be not. Let's look. Unspliced, if it is unspliced, if it is partially spliced, or if it is completely spliced. Splicing, removing of introns and only sealing of exons back. Okay, so primary transcript is approximately 9 kb in length. Unspliced primary transcript and partially spliced transcript would not be exported from nucleus normally, but in case of retroviruses there is a mechanism known as rev dependent mechanism, due to which unspliced and partially spliced RNA can even be exported out. First of all, the completely spliced one will easily be transported to the cytoplasm, okay, and will transcribe and will translate to give proteins TAT, REV, and NEF. Now, this REV protein is responsible for making both of these RNAs export out of nucleus, because these RNAs possess a sequence called RRE in their RNA, that is REV, Rev response element. Rev is an RNA binding protein with sequence specificity. So it will only bind to RRE. It will come and bind to RRE, this Rev protein, and will lead to their nuclear export, and they will be moved on to cytoplasm, same as here, and then they'll translate into their respective proteins. It will give GAG polyprotein and GAG pol fusion protein. It will be giving VIF, VPR, VPU, and ENV protein. Now, we can conclude from this that this completely spliced one is Rev dependent pathway, and both of these are, sorry, this is Rev independent pathway, and these both are Rev dependent, dependent on Rev for the nuclear export. And as the unspliced RNA primary transcript, it can get translated in cytoplasm into its respective proteins, or it can be used again as mRNA, genomic RNA, for packaging of viruses. The length of unspliced one is 9 kb because it is not spliced. Partially spliced one will be approximately 4 kb. That means approximately one splicing is done. Whereas in completely spliced case, more than 30 mRNAs are produced of approximately 2 kb length. That's all for the video. Thank you.